Hello, 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 biologists, and welcome to a quick example video about epistasis. This goes along with your extended practice 4, I think maybe practice 3 as well, and your epistasis lesson. And this is going to be a quick one to show you what we mean by gene control. So here we go. In mice, the agouti, which is a weird color that is always mentioned in biology, but it's just like a grayish kind of speckly type deal. So in mice, the agouti grayish color is dominant over black color. There is a second epistatic gene, keyword, that controls pigmentation. It is Mendelian. In the homozygous recessive case, no pigment is deposited in coats and mice are albino. Then we're going to find the phenotypic ratio for two heterozygotes. But basically, um, take a look at the epistasis presentation that's posted, but it's kind of like an on-off switch. Because like the first gene that we have here is determining the black or gray color, but the second gene controls if there's any color at all. So it doesn't matter if you have the gene for black or gray if you're not having the gene to even have a color. And that's what epistasis really is. It's like you can't express either of these first two genes unless you have a dominant gene for the pigmentation one. Um, so yeah, first thing we're going to do is identify our type of inheritance, and this is like going to work out basically like a normal dihybrid cross, but then just the way that we read the genotypes at the end are going to be different. So I'm going to kind of go through this quickly, but then talk about the difference here compared to what you're all used to. So we determine the inheritance pattern here. First of all, we determine it's like epistatic, whatever, but these are still both just Mendelian traits. So I'm defining them as A and B just because they're nice letters and near at the very, not near the beginning, at the beginning of the alphabet. So we got our agouti. We're gonna have a capital A, black, lowercase, because black is recessive. Similarly, pigment's dominant, so we're gonna put a big B, and no pigment. The like epistatic part here is recessive. So with that in mind, I just said that both mice are heterozygous for both, so we know that means they're both big A, big B, little b. Easy enough. Then they're all gonna have 4 gametes. This is all the same as like the stuff we were doing on the last assignment in the normal dihybrid. And the Punnett square is also like gonna work out the same. It's gonna be your classic that went early. Okay, it's gonna be your classic 9331 with a twist on how we read it, on how we read it. So this would normally be 9331 because we have like just like a dihybrid cross. So we have the normal scenario, except one twist here, which I added this box to be the same as these. We're going to end up with essentially a 9, 3, 4. It's kind of like the way of putting it. Because, here, just before I get to that, if you have the double recessive case here, see like these 4 boxes, you're going to be albino and not have any color. On the next slide, I'm going to show you a picture that actually shows the mice. But basically, these 4 boxes have the exact same phenotype. Normally, these ones would be agouti, right? Because they have like the capital A. But this A allele does not matter whatsoever when you have 2 lowercase b's, because that means you don't have any pigment at all. So this case would normally be like the one lone double recessive case, but it's going to look the exact same as these 3 other cases, because in any of these cases you just don't get pigment. Then the rest is the same, so like these ones will all be agouti and just look like that because they all have capital B's, so they all have the pigment. Similarly, these 3, they're recessive for color, so they're black, but they have the pigment, so they'll look black. But these 4, even though you'd think that they might be agouti based on the A's, they all are just going to look the same albino like phenotype because they don't have any pigmentation to begin with. So basically this recessive gene is controlling the expression of this A gene. When these are recessive, it does not matter that A is dominant or anything because it won't be expressed. So we end up with 9 out of 16 agouti mice, 56.25%, 3 out of 16 black mice, and 4 out of 16 albino. So this is the difference here. These 3 and this 1 end up being the same here in an epistatic example. Here's what it looks like in a really pixelated photo, just like with actual mice. You can kind of envision it. They arrange this a little differently, but like same deal. Oh yeah, they did this like way differently because they use the A's and B's, but basically just like color-wise, we're gonna end up with 4 that are albino, and those are these 4 here in our example. 3 that are black, and the rest will be this kind of like yellow grayish color, agouti. So yeah, the whole thing with this is that the one gene controls the expression of the other. That's why we call it gene control. So if you have the gene here that's saying no pigmentation, it doesn't matter about your A. So this like B gene, the gene for pigmentation, is controlling if the color gene's even able to be expressed. And that's epistasis. So with that in mind, I wish you luck on the best of your practice. Probably get one more video up here. Email me with any problems or like any questions you have about the problems, or any requests for a video you may have. And yeah, have a lovely day, everyone. Take care.